Bence-Jones protein is a small protein made by plasma cells or white blood cells that produce antibodies. It was first described in a patient admitted to St. George's Hospital in London. Dr. Henry Bence-Jones tested this urine and found a substance in it that was precipitated by the addition of nitric acid. Jones proceeded to call this substance hydrated deoxide of albumin. It was first characterized by its peculiar attribute on heating, precipitation of the urine at 40 to 60 degrees Celsius, and redissolving of the precipitate at 100 degrees Celsius. The Bence-Jones protein urine test is used most often if the doctor suspects that someone has multiple myeloma. It is found in the urine of most people with that type of cancer that begins in plasma cells. Healthy urine does not contain Bence-Jones protein, so its presence is an indication of a health problem. Multiple myeloma happens when the plasma cells, which make the antibodies that fight infection, start to multiply uncontrollably and release Bence-Jones protein. This protein does not serve any useful function in the body.
The materials needed are centrifuge, 33% acetic acid, litmus paper, test tubes, beaker, cold distilled water, fresh urine, electric hot plate, and thermometer.
Centrifuge about 25 milliliters of fresh urine. Test for protein with sulfosalicylic acid as in routine urinalysis. If the result is negative, Bence-Jones protein is absent since SSA precipitated this protein. If positive, acidify 20 milliliters of urine with 33% acetic acid using the litmus paper. Place 5 milliliters of urine in each of the 3 test tubes. To the first tube, add 1 drop of 33% acetic acid, 2 drops to the second tube, and 3 drops to the third tube. Place all 3 test tubes in a beaker of cold distilled water on an electric hot plate. Mount a thermometer in the beaker. If albumin and other proteins are also present, this will not precipitate until the temperature rises above 65 degrees Celsius. If a mixture of protein is suspected, heat the test tube having the greatest turbidity to boiling. Filter rapidly while hot, and allow the filtrate to cool slowly. If Bence-Jones protein is present, it will reappear as the filtrate cools to about 50 degrees Celsius.
Interpretation of Bence-Jones protein. If Bence-Jones protein is present, at least one tube will develop turbidity since proteins have an unusual property of precipitating at 40 degrees Celsius to 60 degrees Celsius, and then dissolving when the urine is brought to boiling point and reappears when the urine is cooled.